Ellen.
And I'm Alex.
And today we're going to go over the central dogma and DNA structure.
We'll also be talking a little bit about the problems that can arise during DNA replication and protein synthesis.
So if you look at the structure of DNA, it's made up of nucleotides that are paired to each other, and these nucleotides fall into two different categories.
Purines and pyrimidines. Purines and pyrimidines always match with each other. Purines are a double ring, and pyrimidines are a single ring. And they have to match double with a single because there's not enough room to have four rings next to each other in the DNA strand, and there's too much room for just two.
So the purines are thymine and cytosine, which have double rings, as you can see here, and thymine always matches with adenine, a pyrimidine, and cytosine matches with guanine. And the cytosine-guanine bonds are stronger because it forms 3 hydrogen bonds rather than 2. So a DNA strand made up of mostly C's and G's is going to be more tightly wound than one made of A's and T's.
So in DNA replication, the double-stranded DNA will split. So all of the hydrogen bonds that we just talked about will break between the two strands, and there are the coding strand and the template strand. And DNA replication will always move along from the 3' end to the 5' end of the template strand because the new strand that is being built 5' to 3', just like the coding strand on top.
So here we've drawn an mRNA strand, which, as you can see, is formed by forming bonds with this template strand, and it matches the coding strand exactly, except that the thymines are replaced by uracil in RNA. So that's how you can tell the difference between those.
Yeah, and when RNA is formed, it will leave the nucleus and it will go to the ribosomes where it will form proteins. Proteins are made out of amino acids, and the structure of the amino acids is coded into the RNA sequence. So there's these things called codons, and they are the 3-letter sequences of nucleotides. And so each 3 letters will code one amino acid. And it's important to note that the amino acid code is redundant. So each amino acid can be formed by multiple 3-letter codes.
So, for example, ACU is a codon that codes for the amino acid threonine. But if you switched the U to a C, ACC would also code for threonine. So a mutation that caused that switch would still create the same protein.
And that's the body's way of protecting itself. And if you look at your codon table on your own free time, you'll notice that a lot of the codes that code for the same amino acid are very similar to each other, and that's just, it protects the body from having too many deleterious mutations.
A change in the nucleotide sequence does not always result in the same amino acid. So if you change one of these nucleotides, it might cause an entirely different amino acid to be coded, and that could cause huge changes in the protein and how it folds. For example, changing a polar amino acid to a non-polar will create different bonds in the protein, and the tertiary structure will be lost.
But if you were to go from polar to polar, for example, you'd have to look at that on a case-by-case basis because you don't know how that is going to affect the function of the protein. But if you look here, so this first amino acid here, ACU codes for a polar amino acid called threonine. If you change that middle amino acid to a U, now it codes for isoleucine, which is a non-polar amino acid. And that can cause massive problems in the folding. So one classic case study in basically every biology class you're going to take is the formation of sickle cell anemia, when crucial polar amino acids in hemoglobin are replaced with non-polar amino acids, and the protein completely loses its function and it causes all sorts of health problems.
The cell's defense mechanism against these mutations is DNA polymerase, which scans the DNA and looks for kinks that could be caused by mispairings of nucleotides. So if you bond 2 purines or 2 pyrimidines, it will cause a bulge as two purines are bonded together, seen here, so that's a bulge, and then two pyrimidines bonded together will cause it to pinch in. So if the DNA polymerase sees one of these problems, then it'll excise that part of the DNA and replace it with the correct nucleotide.
You'll run into problems when you swap one pyridine for the other pyridine, or vice versa with the purines, because then it's not as apparent in the backbone of the structure and it's harder to find the mutation, so the body doesn't always fix it, and that's when you get some problems occurring.
Right, so that's the end of our video. Thanks for watching. I hope some of this cleared up DNA replication for you.